too, sorry. A Google form to collect that information as well. So if you'd rather not do it verbally, we'll get to that in a little bit. I'm going to drop the agenda in the chat. If I can find it. Where are they? Okay, yeah. It's also was included in the last email out to everybody. Drop that in there. And I'm also going to drop Brett's paper, which I think has my highlights in it, but hopefully you can ignore those. Those are both documents that will be helpful for our talk today. Before we get going, does anyone have last minute announcements or things they want to add to the agenda? All right, awesome. Well, so I think just a few quick announcements is that the April issue of the transmitter is out. There's a link to that in the agenda if you haven't seen it yet. Lots of great highlights in there, and lots of good video content in the last month. So check those out. For those of you that don't know, because we live in this world from the shoulders up, I am going on parental leave any day now in the next couple weeks. We're having a baby boy due soon. So I'll be out mid-May through mid-August, and Jared, I think so. Jared is going to be, you know, as always, my counterpart while I'm out, but we don't really have a backfill for my position. So do try to, you know, temper your requests of him or your expectations in that time. He's going to be kind of doing two jobs at the same time. So, but I'll see you guys. This may be the last time I see you for a while, but other than that, we can jump into the highlights reel. So I'm going to share my screen. All right. Sorry. Does that look okay? I can't see anyone's face.
Looks great.
Okay, good. All right. So thank you to everyone who contributed to this. This is always actually one of my favorite things to put together for Movement of Life. It's just kind of our way to capture the major highlights. It's by no means comprehensive. It actually doesn't even begin to touch on publications that people have put out, just because it would be overwhelming in this context. So we focus more on conservation achievements, partnerships, awards, things with that are a little more easy to communicate in this format. So we'll dive in. So starting out with conservation achievements, the Scimitar Horned Oryx project released 20 oryx and 25 addax into the Awadi Achim Reserve in Chad. They've introduced, I think it's over 300 animals maybe now, but to date for oryx, and 90 founder addax released to date. And at least 68 wild-born oryx and 19 wild-born addax last year. So those are really tangible, awesome conservation achievements from the oryx team, which is led by Catherine Merides. The Great Plains Science Program also translocated 28 swift fox to Fort Belknap Indian Reservation, where at least 16 kits were born in the wild last year. And the Smithsonian Migratory Bird Center contributed data to multiple management, education, and conservation initiatives. The list was so long, I just had to summarize them here into international shorebird conservation initiatives, 3 national shorebird conservation initiatives, and then Audubon's Migratory Bird Explorer. So SMBC is a key partner in that data set. The Conservation Ecology Center and partners completed the first digital aerial and satellite-based wildlife surveys of the greater Mara ecosystem in Kenya. So that's Jared and I were leading that with our partners in Kenya, and really pushing on AI-based methods for detecting multiple large animals in aerial imagery. We're hosting a workshop for that coming up in September to share results. The giraffe team, which is led by Jared Staubach and the Giraffe Conservation Foundation, is trialing new low-cost, low-impact CERES tags. Chris Fleming, as always, is as many publications. One of I think a couple of the biggest I've cited here for last year. Developing very user-friendly software to improve the accuracy of habitat suitability models and population level home range estimations. So there's links to those papers there if you want to check them out. And in Matt Ogburn's team at CERC, Michelle Edwards discovered new bull shark nursery habitat in Florida. And that was written up in maps. NZCBI, Smithsonian Migratory Bird Center, and Department of Animal Programs and Reproductive Science finished transmitter trials for the reintroduction of Guam kingfishers. This is a great photo of Amy here. And then last year, there was a really great 3-part series from the Smithsonian Women's Committee that was celebrating women in STEM. And we had multiple movement of life scientists featured in that. There's a link to the recording there if you'd like to check it out. It was for Earth Day last year. That included Mel Songer, Catherine Meredith, Autumn Lynn Harrison, and Kim Ritchie. And as usual, Chris Fleming and colleagues hosted a sold-out movement modeling workshop at the Wildlife Society's 2022 annual conference. So really trying to work with managers and build up the quantitative capacity of wildlife managers to work with animal tracking data. Movement of life reps. So Autumn Lynn, Jared, Peter, and myself attended the Internet of Animals Symposium at Yale this year. It was really, it was a unique and encouraging experience, I think, overall. Just getting to NASA's kind of re-engaging in the animal tracking world and seeking input on what type of hardware and software and just space-based technologies are needed to really propel the world of animal tracking forward. So it was a cool thing to be part of that. Here's Martin Benavides at the, I guess it's the Sharks International Conference. So it's and he presented on their collaborative project with CERC on elasmobranch tracking. And same thing, Matt Ogburn's Fisheries Conservation Lab hosted the Atlantic Cooperative Telemetry Network training workshop. So that was Kim Ritchie and Matt Ogburn in collaboration with the Ocean Tracking Network. And overall, I actually don't think this is a full number. We should try to update this maybe before it goes around to add administrators, but we've mentored at least 13 interns and advised 4 research fellows in the last year. Another big activity last year was the Earth Optimism Folklife Festival on the National Mall. This is a photo of Ellen Stofan speaking with Irene Amoke of Kenya Wildlife Trust, one of our key partners in Kenya. And we had several partners from Kenya, One Mara Research Hub, and the sorry, blanking on their name, but the MA Trust also came out and joined our group on the mall and had a booth to speak about conservation in the Masai Mara, and with a focus on habitat connectivity. Catherine Meredith also moderated a panel on saving grassland species. Some awards that our teams received this past year include an honorable mention at the Better Satellite World Awards for Movement of Life. Iridium nominated us for that award. Andrew Jakes was awarded special recognition at the 29th Biennial Pronghorn Conference. And NIFWIF awarded $5 million to 4 tribal communities and the Great Plains Science Program to support bison restoration and enhance landscape connectivity. This is kind of a roundup of major media coverage received. So it's best to check this out, click on the links. And if there are links missing, there's a couple of links missing here. If anyone would like to add those before we send this up to leadership, please reach out to me and I'll be sure to do that. Lots of new tracking initiatives in 2022. And lots of collaborative new tracking initiatives. So I think one of the most collaborative ones is this one with the Migratory Bird Center, Virginia Working Landscapes, and Great Plains Science Program launching a new range-wide tracking study of both eastern and western meadowlarks. The Center for Conservation and Sustainability and Cirque Fisheries Conservation Lab launched a new study of elasmobranch movements in coastal Peru. That's Martine's project. And NZCBI's Reptile Conservation Ecology Lab began the first known study of juvenile wood turtle movement and survival in Virginia. Cirque Fisheries Conservation Lab also began tracking alewife migrations in response to climate change. I know they've been still busy out tagging this year for that. Began tracking predators of Sarasota Bay dolphins with Moat Marine Lab. And Virginia Working Landscapes and Smithsonian Mason School of Conservation launched the first automated tracking study of grassland birds during their vulnerable post-fledgling life stage. And that is happening kind of in our backyard of Front Royal. Some exciting future directions to keep an eye on this year is that Joe Klowski of SMSE will be tagging 20 kestrel pairs, so 40 total kestrels at nest boxes in Central Virginia to study sex-based variation in habitat selection and behavior. So this will be a really cool comparative study of how males and females that are paired are splitting resources and time. NZCBI's Reptile Conservation Ecology Lab will deploy 50 miniature GPS units to record fine-scale movement and survival of juvenile wood turtles. And Virginia Working Landscape plans to install 42 automated tracking nodes and deploy 85 tags on grassland bird fledglings this year. So busy year coming up. I also want to highlight our new collaboration with Iridium that's supporting or expanding 5 tracking projects worldwide. All the species listed there, including bobcat, which Brett Jesmer is PI on that project, and his graduate student Nicole Gorman has been leading field efforts. I think you've got 6 at the end of the season, right, Brett? Is that, yeah?
Yes.
Yes, six tagged. Out in Albemarle County, Virginia, looking at habitat connectivity. And then the Meadowlark range-wide tracking project will add 8 new sites and collaborating institutions, including 2 in Canada. The Bird Friendly Coffee Program has also had a great year and is going to be tracking 4 migratory species that overwinter on Colombian coffee farms. And the Fisheries Conservation Lab will work with the Atlantic Cooperative Telemetry Network to develop a regional strategy for evaluating responses of migratory marine species to offshore wind energy development. And sorry, what is what's CCS? No one's reminding me. Okay.
Conservation and sustainability.
Center for Conservation. I always want to mix you up with CSR. Center for Conservation and Sustainability will expand its acoustic receiver array off the coast of Peru. And this is also a collaborative project with Matt Ogbird's group at CERC. So just a final wrap up to say a major congratulations. You guys, as always, have just achieved more than would be feasible with the amount of resources we have at hand. So I hope everyone can take a little time to reflect on accomplishments and highlights from the last year. And just thank you all for contributing to this presentation. There will be time, I mean, I'd say if you have seen this and would like to make any edits or additions before this goes up to leadership, please reach out to me in the next week and I'll make sure to do that. But otherwise, we're going to send this on up so everyone can know how awesome you guys are. I'm going to stop this. Oh, great. Thank you. Okay. Any questions or follow-up while we're all here together on that? Okay.
Thanks for pulling that together, Lacey.
Yeah,
It's really impressive to see it all in one like that.
I agree. Okay. So nobody submitted anything for new or departing staff, but is there anyone that would like to be introduced? Yes, go Autumn Lynn.
I don't want to be introduced, but I want to do an introduction because our new staff member, Jessica Howell, just received her SI email. So she's official now and on our network and attending today. Jessica is joining us from the American Bird Conservancy and also NRCS to support the Shorebird Science and Conservation Collective direct conservation projects using contributed tracking data. So Jessica, welcome. And I don't know if you want to introduce yourself to the group, but.
Hi, thanks. Yeah, I think this is my 4th week, so I'm excited to be on the network. And yeah, I was working out of Northwest South Dakota on private lands with grassland birds for the past 4 years. I'll be based out of Eastern Montana and excited to kind of get back into the shorebird world. So, and work with partners here in the Northern Great Plains and Prairie Potholes.
Welcome, Jessica.
Anybody else? Any new interns or fellows?
We have new interns. Itai.
Yeah.
Itai is on the call. Itai, if you're not embarrassed, you can turn on your camera. She's a new intern who's going to be helping with some of the work that James is going to be talking about today. She's working on several projects, including our bison movement project, our prairie dog movement project, and swift fox reintroduction.
Okay. Hi, Itai. Welcome.
Hi, everybody.
Anyone? And it's Kirsten. She's not on.
No, she's there's another intern, but she's not on today. Yeah.
All right. Anybody else? Okay. And departing staff? I don't, no one that I know of at this point, but okay. Well, with that, I would love to turn over the majority of the rest of our time to our speakers. So Dr. Brett Jesmer is here with us from Virginia Tech. He's an assistant professor in wildlife ecology restoration and conservation lab. And we've been working together for the past, I guess about the past year on this bobcat tracking study in Virginia. And, you know, Brett, I just want to point out, was really enthusiastic about using this opportunity to really identify new collaborators at Smithsonian. So if you're excited or interested by what he's speaking about, please follow up with him. And he and Nicole, his PhD student, will be on campus May 9th and 10th. I've sent an email around to a core group of people, but if you didn't receive that and would like to meet with him or Nicole, also reach out to me or him, and we'll make sure to get you on that schedule. So, and he'll be at the Virginia Working Landscapes meeting on May 9th as well. So, Brett, you're going to be speaking about how animal tracking is moving in community ecology forward. So opportunities and challenges. So I'll let you take it away with screen share.
Okay, thank you, Lacey.
Yeah.
So let's see. Take me a second, just please bear with me. And while I'm getting this set up, I just want to thank Lacey for the opportunity to join you all today and talk about a little bit about some of the research I did a few years ago and I'm still engaged with to a fair degree. And this was a paper that some colleagues of mine during my postdoc at the Max Planck Yale Movement Center, which Lacey mentioned earlier, hosted a Internet of Animals symposium, I guess, apparently over the past year. And so that was with Dr. Walter Yetz, who's the PI of that lab, and my collaborators, Remington MOL at the University of New Hampshire, and Raul Costa Piera, who's at Campinas in Brazil. And so what I'm going to talk about today are how animal tracking data can advance our understanding of community ecology, but there are many opportunities that I'll identify and challenges. So before I even get started, I want everyone to recognize that what I'm going to present today isn't in any way sort of a representation of how movement data is going to be a silver bullet for solving all of our challenges in community ecology and deciphering inter and interspecific interactions. But it's just a framework to get us thinking about how we can use movement data to maybe hopefully advance some of these questions. And I'll spend, can I advance slides here? No, got to click on one of my screens. There we go. So as you see at the top here of today's talk broken into sort of 3 segments. Now, setting the stage is actually going to be about half of the talk, and then I'll spend about a quarter of the talk talking about opportunities and another quarter talking about challenges. And so I really want to spend some time on setting the stage because understanding how the niche concept and community processes are related is sort of central to understanding how movement data might help advance some of our understanding of inter and intraspecific interactions, which are at the core of community ecology. So first, I want to talk about niche partitioning and the competitive exclusion principle. So essentially, the competitive exclusion principle, sort of set forth by Gauss in 1934 and later sort of solidified by MacArthur and Levin, simply states that to coexist, 2 species cannot occupy the same niche indefinitely, right? And I think many of us are probably aware of that definition, but essentially 2 species can't occupy the same niche at the same time. One would outcompete the other essentially over time so that they could not coexist. And we refer to this as the Grinnellian component of the niche, right? So essentially the Grinnellian component of the niche are, when we think, are the aspects of the niche that involve resources, right? So habitat types, food types, and that sort of thing. And we usually refer to these things as horizontal, or we study these things in regards to horizontal communities. So, right, like clades of herbivores, which I'll demonstrate here with deer and elk, and I'll dive into this figure a bit more in a moment. Carnivores, frugivores, granivores, et cetera, and how they might be competing for similar or shared resources. So what I'm showing you here in this figure is just a heuristic that I drew out of potential deer and elk resource space and how they might partition that niche. And then you can see each of these solid bell-shaped curves as an individual, orange being deer, blue being elk. You can see they're doing quite similar things. And then at sort of the peripheries of that niche space, if you will, they're overlapping, and there's some opportunity for interspecific interactions and competition to be occurring there. So one thing I'll sort of plant a seed for before I move on to the next slide is that there's now a well-established literature and a lot of empirical evidence demonstrating that individuals vary in their resource use. So they vary in their Grinnellian niche space within a population, and certainly in widespread species with large geographic ranges, individuals and populations might do very different things in different places. And even though that's well recognized, we still really focus on populations or species as the unit of interest in community ecology, right? We don't really focus on individuals as the unit of interest. And I mean, some people are doing that. It's definitely catching a little fire, but largely we're still focusing on populations as the unit of interest. And just, as an example, in the last 5 years, we have some papers coming out of Rob Pringle's lab at Princeton with postdocs and graduate students, Kartzenel and Pansu, these papers from PNAS and Journal of Animal Ecology, using DNA metabarcoding to, for the first time, really sort of nail down how species in the Mozambique, I was thinking, is this Serengeti? No, Mozambique, are partitioning the dietary niche, right? But again, we're still at this point focusing on species, and this isn't a dig on the Pringle lab by any means. They do amazing work, and they also have several papers looking at individual variation in resource use that have come out just in the last couple of years. Okay, so to move on to the other component of the niche that I'd like to talk about today is the Eltonian component of the niche, and which is essentially just, sort of been more generalized in recent history to refer to the impacts one species has on another. Traditionally, it was thought of more about as like the ecological role of a species in an ecosystem. And so you have things like primary consumers that are, herbivores or frugivores or granivores. You have secondary consumers, and then you have your, tertiary consumers, which are your sort of your apex predators. So we often think about the niche and a species niche space as the interactions they have with the physical interactions that they might have with other species. And this really focuses on, the vertical community dynamics, that is the consumer resource interactions occurring in a community. So what the paper tried to address was simply how might we use movement data to overcome the challenges that ecologists have had with integrating both the Eltonian and Grinnellian aspects of the niche, simultaneously, right? At the same time, the same place, the same species, quantifying both of these pieces at the same time. And we thought maybe tracking data can help overcome some of these challenges. But before I dive into how movement data might be able to help us overcome some of these challenges, I want to talk about the ecology of individuals, because in the paper, that's what we really identified as sort of the key part of how movement data can help advance understanding of these factors. So, as you all know, as being part of this group, when we're studying movement ecology, we're putting tracking devices on individuals, and we're tracking all sorts of, we're tracking their movements, which help us then relate their movements to all sorts of aspects of their environment. But in community ecology, individuals are traditionally just sort of the sampling unit upon which population level or species level parameters are estimated. There's no or little explicit attention paid to the individual itself or how individuals may vary in their niche space or resource use. We typically just sort of take that individual variation and relegate it into some sort of measurement error, right? So we have a population level estimate with a standard error or a confidence interval. But again, there's very little explicit attention paid to the individual. And just as an example of this, right, think about when we conduct resource selection functions. We usually produce a map. It's a population level response that sort of, illustrates the selection strengths of various parameters in the model. And, we will sometimes, hopefully, putting in a random effect, a random intercept, and even a random slope of individual to account for that individual variation in the model, but we don't explicitly model how different groups of individuals may be utilizing the landscape in different ways and important ways potentially. And so, I just want to take a step back real quick and talk about individual ecology and variation, right? So the theory of natural selection is predicated on the notion that individuals vary in important ways, right? That help them adapt to changing environmental conditions or other pressures in their environment, such as competition. And, ethologists and evolutionary biologists have focused heavily on individuals and how they vary for, over a century now. But in the ecological literature, we sort of have ignored it largely. But in the 1960s, 70s, and 80s, a few different hypotheses were presented that pay either explicit or implicit attention to how individuals vary. And those are things like the niche variation hypothesis by Van Valen in 1960 and by Roughgarden in the 70s, Fretwell and Lucas's ideal free distribution, Pulliam and Holt's ideas about source sink dynamics. And then again, it seems like those ideas kind of fell out of vogue until in the early 2000s, Bolnick, Araujo, and others really picked up the niche variation hypothesis and were testing how individual variation and resource use can lead to speciation and local adaptation and these types of things. And then we also have Gosling's sort of first formalization of the idea of an animal personality. And myself and colleagues, Erica Stuber and Ben Carlson, just last year sort of published a paper in behavioral ecology expanding this idea of animal personalities to spatial personalities, things like consistent individual variation in resource selection or movement parameters. And so, to give you a better idea of this idea of the niche variation hypothesis and how individuals may be varying in regards to their niche space, I have this conceptual figure I want to walk through real quick. So the niche variation hypothesis, or NVH, can sort of be contrasted with classical ideas of the optimal foraging theory, so OFT, where under when resource abundance changes from so when per capita resource abundance, so competition for resources is low, we expect the total niche width, or TNW, to look like the salmon-colored bell-shaped curve there. But as population densities relative to per capita food availability increases and competition increases, we expect individuals to start using sort of less optimal or secondary resources, and you would expect the total niche width to expand out and become broader, niche breadth to increase, as illustrated in the teal curve here in both the panel A and B. Now, under optimal foraging theory, we expect that all individuals would increase their niche width. So that's this within individual diversity. So all individuals would have more diverse diets under increased food limitation. And the among individual diversity, or AID, would be small because they're all doing similar things. So AID is essentially the distance between the peaks of these curves. Under the niche variation hypothesis, we expect the AID to increase, and so that there's a large distance between the peaks of these curves, but the within individual diversity piece to be narrower, because essentially individuals are specializing, groups of individuals are specializing on different aspects of the dietary niche in this case, or habitat or resource niche, such that they're partitioning that niche space and reducing competition. And this review in panel C by Bolnick et al. In 2003 showed that about half of the time in nature, the response to increased competition and food scarcity follows the assumptions of the optimal foraging theory, and about half the time follows the assumption of the niche variation hypothesis. And so we tested this in 6 different populations of moose in the Intermountain West that varied in their per capita food abundance, which is here on the x-axis. So essentially more food, so less competition here on the right-hand side of the x-axis, and food scarcity or less per capita food availability on the left-hand side of this axis. And what we found was in accordance with both the niche variation hypothesis and the optimal foraging theory, that total niche width did increase as food abundance became more scarce, and was much smaller when food abundance was high. But that expansion in total niche width was primarily driven by increases within individual diversity, right? So under resource limitation here, you had wide bell-shaped curves for individuals in those populations. So essentially what we found was that in that study, in that system, that moose were responding to resource limitation in accordance with optimal foraging theory, and that their niche breadth changed as it is here in panel B. Okay, so what this means is that niche breadth is plastic, right? It can change over time in response to resource limitation. Because we often think about, right, that niches are just this sort of static thing, like you measure it and that's what it is. But this study sort of illustrates that they're plastic and they can change over time. And so if we play this out over time, and now we expand this from a one species study to a potentially two species study, again, I'm using this deer and elk heuristic, you can see that over time, as resource abundance changes and ebbs and flows, if these niches are expanding and contracting, that there's certain periods of time where these species should be competing more for shared resources, and certain periods of time where they should have less competition and less overlap in their niches. And so then if we play that out even further, we can see that niche variation can alter inter and intraspecific interactions. So what I just showed you was, this plasticity of the niche, which should change over time, what we're seeing here on the left. But we should just generally see the classical sort of prediction that intraspecific competition will generally be greater than interspecific competition, as illustrated by the thinner lines between these two species and the thicker lines within the species. But in those other 50% of the time that Bolnick demonstrated that the niche variation hypothesis is what drives expansions of niche width under resource limitation, we should expect that actually sometimes interspecific competition, interspecific competition, like this line, could be much stronger than intraspecific competition, which sort of alters the way we think about how species coexist in a community. Okay, so now I'm going to bring you back to the actual paper that Lacey shared with you all, and we tried to distill all of this into one, what one of my co-authors likes to call a hero figure, and it's a lot, but it's pretty cool. So it'll take me a minute here to walk through this. So those of you, I guess all of you in this room, have done some tracking data, some tracking studies in the past. And so in this case, we have 3 species of birds, multiple species or multiple individuals of each of those species, mapped out their tracks over the landscape here. And if we were to annotate those tracks with different environmental, remotely sensitive environmental layers like NDVI, land cover, topography, etc., we could then look at how their use of these different Grinnellian factors changes over time, and how that changes their interactions with other members of their species, so their intraspecific interactions or their interspecific interactions. And we can do this in multidimensional space. This is reduced to 2 dimensions, but there are methods out there to do this in a more hypervolume framework, in accordance with like the Hutchinsonian hypervolume, so that you can then take these interactions in this interaction space, and you could map it out in a topology like we see up here. This topology is with reference to this figure, but you can take this multidimensional resource space and create a topology from it, which I'll show that more in a few slides here. But these are the Grinnellian factors. But at the same time, using that tracking data, We can look at pairwise spatial proximity and different interactions, and I'll talk a little bit more about this later as well, to look at those Eltonian interactions, right? So avoidance, attraction, neutrality. So like territoriality, right, between two individuals of the same species would be an avoidance behavior, and we can detect that from movement tracks. Mating would be an attraction behavior that we might see within members of the same species. And then we might also see predation, where there's attraction, interspecific attraction from one species, i.e. predator towards prey, and an avoidance of the prey of the predator. And we can, detect all of those things at the same time simultaneously as we're quantifying these Grinnellian factors. And so in this way, this is how we sort of envisioned movement tracking data, sort of helping to advance our ability to simultaneously quantify Grinnellian and Eltonian factors in a way that will help us sort of disentangle community dynamics better. Okay, so to build off of that, I created this heuristic. So on this left-hand panel there is essentially this bottom right-hand panel that I'm showing you here in this Grinnellian environmental space. And so again, we have multiple individuals, multiple species in a community, with the solid lines being the individual niche space, Grinnellian niche space, and the dotted lines being the population level niche space. And under classical ideas in community ecology, we might create a topology, again, at the population or species level, as illustrated in the middle, or when individual variation is either actually weak or assumed to be weak, versus what it might look like if we actually incorporated individual variation in Grinnellian niche space into that topology. You can see that there's like certain individuals of elk, for example, these two sort of phenotypes, these two elk phenotypes interacting with deer, and these two phenotypes interacting with hares. But these other phenotypes have no interaction with deer or hare, right? So unlike this middle panel where all elk interact with deer and hare. So this is where we might then start to be able to understand what the sort of key phenotypes and traits and roles are that different species are playing in a community. And so we can also then extend this to a multi-layer interaction network using multi-layer network analyses, where again, here on the right, we have deer, elk, hare, and foals are my examples interacting and what that topology would look like. And then we have two carnivores like bear and coyote here in this simple topology in the middle. And if we then sort of connect these two things up, oops, there we go, connect those two things up, we have a ditrophic carnivore herbivore multi-layer interaction network. And so you can see now how movement data is starting to allow us to look at and integrate both the Grinnellian and Eltonian factors, right? Because now we're looking at things like predator-prey interactions at the same time that we're looking at competition amongst or within an herbivore guild. So this is one way that we can start to, I hope, maybe like advance our ability to quantify all of these community level interactions. And then we can add in things like DNA metabarcoding, for example, to look at the interactions with all of these herbivores in this horizontal herbivore community with the plants that they rely on to create this tritrophic, oops, that's a typo at the top, that should be tritrophic, not ditrophic, carnivore herbivore plant interaction network. Okay. So those are that's, I just got through the opportunity phase of the talk where I think we've identified some ways that movement data can help drive community ecology forward, but many challenges remain. And I don't think tracking data is a panacea for integrating Grinnellian and Eltonian factors because, this is a non-exhaustive list, but tracking devices at this point are mostly available for medium to large species. So we're not going to be able to do much with the smaller bodied species. And I don't think movement ecology can really contribute much here. This is more of an engineering problem. we usually cannot track all individuals in a population or even at a study site, right? So we're going to inevitably be missing interactions amongst individuals. I do think movement ecology and models can help alleviate some of those issues, but probably not perfectly. Even fine scale data, like a 30-minute fixed rate on a GPS color, is likely to miss interactions, both inter and interspecific interactions. And then, as I mentioned, we need auxiliary data to quantify interactions with things that we can't use tracking data to quantify, such as interactions with plants and insects and those types of things. And then lastly, and I think this is a big thing, is that we need demographic data to accurately assess Grinnellian niche spaces, right? Because, the gold standard in niche modeling is that in order for a set of environmental conditions to be considered part of the niche, you have to demonstrate that that niche space can sustain a population relatively indefinitely, right? Like, if that population has a declining growth rate, right, lambda less than 1, that's not part of the niche. That's like a sink, right? It's not part of the niche. So we need demographic data to support these ideas of the Grinnellian niche. So I'll briefly, just to wrap up, talk about these two aspects. Like I said, I think movement data can help us, movement models can help us overcome point 2, but I'm going to just talk about point 3 and 5 real quick. So what I'm showing you here on the bottom left is a heuristic from Nathan et al. 2022 science paper, where you have 2 species of birds. You have their true path in this sort of thin gray line, squiggly line moving through the landscape here. In the blue, it's actually pretty tight to the sample path, right? So this is like you have a fixed rate every 15 minutes or an hour or whatever, and these are the GPS locations you're getting as it's sampling that path. And so where I think movement models that have already been created, already published by Chris Fleming and Schlegel, for example, is using time series kriging to reconstruct those paths to create occurrence distributions, which do a much better job at approximating that path, right? It doesn't recover it perfectly, but it does a much better job at recovering that path. And now, instead of just the old school connecting points together with lines, we are actually able to pick up some of these important interactions, like you can see where the lines intersect with each other. And then if we add in step selection functions, you can essentially ask questions about attractance and avoidance between pairs of individuals by essentially creating an occurrence distribution, such as this dotted line here, and asking if this individual in a step selection function framework is selecting for or against or not at all for the path of that individual. So if there's no selection for or against, you sort of this neutral interactions. In this case, blue was attracted to red, but red avoided blue. So this might be like a predator-prey scenario. And then in this case, you had 2 individuals attracted to each other. So this might be social conspecifics or a mating event or something like that. Oops, going the wrong way. Okay, this way. There we go. And then lastly, just to wrap up, so as I mentioned earlier, if we do have a situation where you have individuals specializing on different subsets of a environmental niche, but there's only one essentially group or environmental space that supports that can sustain a stable or increasing population growth, so lambda equal to or greater than 1. Now, this would actually be the only true niche space for that population or species. And then if these 2 sort of peripheral, variants of the niche had population growth rates less than 1, then these would just be sinks, right? So this would be the only actual Grinnellian niche space, and these would just be sink habitats or sink resource spaces. And so we really need to be able to do that to better quantify Grinnellian niches. And using movement data, there are some ways we can do this. And I'm from my experience and talking with people in the field, this works for many species. This doesn't always work for all species. It's kind of dependent on their movement behavior and their reproductive behavior. But this is an example of using step length distributions, and different types of models fit under an AIC framework to detect when an individual had no calf, right? So during parturition or just post-parturition, the movement rates of mom decline dramatically. And then as that calf grows, they start moving more and more over time as the calf becomes more mobile. But in the case where a calf, there's parturition event, calf is born, and then the calf dies for whatever reason, whether it's predated or dies of malnutrition, then suddenly you have this big jump in this increase back to the standard for pre-parturition step length pattern. And so we can use, moving windows and behavioral change point analyses and AIC model selection to sort of identify these different events and quantify parturition and neonate and juvenile survival. And then, of course, we all have mortality sensors on our collars, so we know if the mom survives. So we can use that information to then quantify the vital rates and look at population dynamics and demography of these species and attribute those to various niche spaces. And so with that, I just want to wrap up by acknowledging all my co-authors at the University of Wyoming and at Yale and other places, and our funders for this work, which came from the Wyoming Game and Fish Department, a DDIG from the NSF, NASA, Virginia Tech, and the Max Planck Yale Institute. And with that, I'll take any questions, and thank you for your time.
Thank you so much, Brett. We have plenty of time for questions, so if anyone wants to jump in, you can type it in the chat or just ask. Autumn Lynn, go ahead.
Hey, Brett, thanks. That was really, really interesting. And I was thinking a lot about subbing in the Alaskan tundra food community into your plots and trying to predict what I would see there. One question I have is, when you think about niche change through time, are you thinking about that still at the population level, or also thinking about interannual variability across individuals? How do those dynamics play out of tracking individuals across multiple years versus the population stability of its niche over multiple years?
Yeah, that's a great question. You can do both. You can work at both the individual level and then scale that up to the population level. But with tracking data, we're inherently tracking individuals and using those individuals to sort of scale up to some sort of population level inference. But I think, as I mentioned in the talk, that it's important that we think about the individual variation and the ecology of these individuals and how their resource use and niche spaces may be changing over time, and how that essentially, again, scales up to the sort of population level patterns we're seeing. Because it's actually the individuals that are interacting with their environment and with each other, not so much the populations per se.
Yeah, thank you. I feel like a lot of times when I've been looking at this, I'm limited by sample size to scale up to the population, but I can at least make inference about what that individual might be doing across years, but never enough data across individuals that have been tracked for multiple years to get that.
Yeah, and that's the big challenge, right, that I mentioned earlier, is even with medium-sized species, we can only set a fixed rate of a couple of hours, a few hours to get maybe 1 year of tracking data. But we usually want to get 2 or 3 years of data to look at that interannual variation. And so that's, a big challenge here, is that hopefully the engineers kind of figure out for us, right? Is like miniaturization of batteries and computer, circuit boards and that sort of thing, such that we can get tracking devices on smaller species and the tracking devices of medium-sized species to last longer and that sort of thing.
Thank you.
Meredith, go ahead.
Thanks, Lacey. Thank you, Brett. Those ideas are super exciting to see kind of all in one place. I had a question about how you think about capturing the individual variation pre-collaring, and whether that is reflected in kind of selecting individuals across the range of environmental variation and assuming that that might be then reflected in the niche space. Or yeah, just I guess your thoughts on whether the describing that individual variation has emerged after looking at the data, or if it's something that you're strategically trying to capture in your collaring plan.
Yeah, that's a great question. I think oftentimes people are just taking what they can get and they're collaring, trapping animals and deploying collars just the best they can without really creating a strong a priori sampling design and thinking about these things beforehand. I do think that's really important though. And that's something that like Nicole and I are talking about with the Bobcat project. And we've spent a lot of time trying to figure out how we can sample in the hopes that we recover a lot of more individual variation. And that's what we're doing at this point. I guess the short answer is I don't have a great answer, but at this point, we're looking at the landscape more broadly and trying to sample individuals across a gradient of essentially habitat types and that sort of thing to try to uncover that variation. And then there's a variety of sort of statistical approaches you can use to control for different confounding variables and that sort of thing when assessing variation.
Okay, thanks.
Peter, go ahead. Yeah.
Hey, thanks for this talk. Actually, my head is still spinning a little bit. I think this is really intriguing, but I guess I have two, as I'm trying to wrap my head around this, I have two main questions. So in some ways, you're really attempting to expand the complexities to make more appropriate models, right, of how species respond to an environment and each other. But how do you, I mean, there's so many interactions, right? How do you decide which species to include and how many you need?
Mm-hmm.
You know, I mean, or which of these relationships are really important? I mean, a lot of what we do is about trying to simplify, right? So we can derive something. That's the first part of the question. And the second part of the question would be beyond this really interesting and important expansion of theory. Where do you see as the application for conservation, for example, or management?
Right. Yeah, all great questions. So first, I think there's three questions there, with the first one being, how do you choose which species? And that's tough. You know, you would have some pre-existing information about the community ecology in your system. And if you don't, that's tough, right? But oftentimes, again, at least with the tracking data component of it, you're going to be sort of hamstrung a little bit in the sense that you can only put tracking devices on certain members of that community, certain species. And then you can think about how you can use some of these other auxiliary data types like stable isotope analysis or DNA metabarcoding or camera trapping or whatever to try to fill in some of those gaps and assess interactions. But so I don't have a great answer. Again, that's why I said this isn't a silver bullet. There's a lot of challenges that remain here in trying to figure this out. And then the second part of your question was about simplification versus generalization. And yeah, I do understand why for decades we've stuck to these more general models where we look at populations or species as the unit of interest. And it does add complexity to dive into the individual level, but I do think it will help us sort of distill down these interactions a bit better. And then I'm sorry, I forgot the third aspect of your question. Oh, how does this apply to conservation? And like, how might I foresee this like applying to conservation? You know, I actually haven't thought about that a ton at this point. Been more thinking at the sort of basic science level and what this means for community ecology. So I don't have a great answer off the top of my head, but with some time, I'm sure we could probably think about some ways to apply this to conservation and management.
That, no pressure on James, but that's some of the hope of his talk, is to share how some of these methods and ideas are being put into.
Well, one idea actually that I have thought about, I'll just throw one idea out there. Actually, a couple just came to mind. But like when we think about, one thing we do all the time to help managers understand, like, what are the important habitats that they need to think about conserving or restore to support a population. And if, again, we just sort of take all that individual variation and just relegate it to a confidence interval or something like that, we may be missing, and we don't connect that to demography, we may be giving managers the completely wrong information, right? So if you have, let's say, a situation where you have two phenotypes, spatial phenotypes, you have two sort of groups of individuals that select for very different types of habitat, and then you just sort of meld them together. Let's say one avoids forest strongly and one selects for forest strongly, and you throw them into the same model, you'll probably get a lack of selection for forest, period. When in fact, if you look at these individuals as essentially groups of specialists, you might find that they're doing very different things. And if you were to just add those models together, you might get a different answer. I'm not sure. But also then if you then expanded that to look at demography, you might find that one of those variants, right, one of those groups of specialists, may be a sink population. And you really want to focus on essentially the source, if you will, or those Grinnellian niche factors that support population sustainability and growth. And those should be the factors that you're giving to a manager to say, this is the habitat you need to restore, or conserve, or protect to maintain this population. Yeah.
Bill, you want to go ahead?
Yeah, Brett, it's very interesting. I'm just interested that for food webs or for some resource selection, you have to know the abundance of whatever you're feeding on. And I don't know how from the movement data, you can see this animal is interacting with this animal, but you have to somehow know, if they're selecting that prey, the abundance of that prey at that underlying level, be that an herbivore or a plant species. And I don't know if you've thought about how can you deduce abundance from the movement data, or do you need some other data set that gives you that kind of information?
Yeah, so this is a really hard thing to do. So if you're looking at land cover, of course, you can quantify use and availability, no problem. But if you're looking at the diet level, right, you're not going to get that really from remote sensing data or from the movement data itself. And that's where that auxiliary data comes in, right? Like diet analysis. And then things get more challenging at that scale, right? That fourth order of selection scale, where you have to go out and quantify availability of all these plants. I'm thinking about herbivores most of the time, so I'm talking about plants right now. But that's a lot of effort to do that.
Anna? Oh.
Right, time and money to do that. People have started to integrate like step selection functions with on the ground vegetation sampling, where they look at the used points and the available trajectories that they created in their step selection models, and go out and quantify, do like quadrant plots and that sort of thing at all of those sites, which is a huge amount of effort. But people have started to do that, but that's certainly a limitation here.
Yeah.
Right. With that, we're gonna move on to our next speaker. Thank you, Brett. And if you have more questions, we have time in the group discussion to talk about those as a group. So write them down and bring them up then. Okay, James, are you ready to go?
As I'll ever be. Yeah.
Excellent. Okay, so James Redcliffe, he is a research assistant at Swansea University in the Swansea Lab for Animal Movement. And he's a current collaborator with Gila Shimon at the Great Plains Science Program, working on black-footed ferret and prairie dog interactions. So I was hoping and that this might be a bit of an exploration into what some of the conservation applications of some of the ideas that Brett was talking about are actually how they're actually being put into action on Smithsonian projects. And also, James, I think speaking more broadly about just how we might use fine scale animal tracking, how to first analyze fine scale animal tracking data, and how to also apply that to conservation questions. Let you take it away.
Cool, thank you. Yeah, so I'm James. I'm from Swansea University, and today my talk isn't necessarily as sort of has a nice slick narrative. It's more of like a scattergun showcase approach about the sort of data we collect at our lab and what sort of analysis we can use to actually utilize that data to draw conclusions and information. And then I'll try and draw some parallels of how we can use that to inform conservation. So yeah, I come from Swansea, and the Swansea Lab of Animal Movement was founded by Professor Rory Wilson, but we almost have like a trinity where we also have Professor Emily Shepherd and Professor Luca Borga. They all work on different animal movement, but they mainly use technology developed at Swansea University. And I've been lucky enough to work on a range of species, and the lab has slowly but surely collecting bits of data from all sorts of animals, because all sorts of animals move in different ways and we're interested in that. So that ranges from whale sharks to giant tortoise to tamarins to cheetahs to lions, but also it's worth noting that Professor Wilson specializes in penguins, and that was the whole idea of using tagging, and I'll explain that in a bit. And then also Professor Emily Shepherd works with birds, so we've tried to get our loggers really small, really lightweight, and I'll explain a bit more about them later. So the idea, just to give you a little sort of analogy, is Rory during his PhD was on some island off the coast of Africa where he was there to just study penguins, and he remembers watching them and observing them, trying to catalogue their behaviour, and they would just go out to sea. And he was thinking, oh my, like, how am I going to understand these animals when I can't even watch them when they're doing their most important behaviors, when they're swimming, foraging, and then coming back to the nest. So he knew he had to get that information somehow, and that's where he came up. Well, he didn't come up with the concept of archival tags, but he started to apply archival tags to animals or to these specific species, and eventually that developed into gauging pressure from weird lead paints and all sorts and syringes, to then actually developing the daily diary. Oh, my screen has gone blank. There we go. And that's what I'll be talking about today. I'll be talking about that daily diary, what sort of data it collects, specifically how it collects that information, and what we can do with that information to answer what the animal's doing, i.e. the behavior. We can get a better idea or estimate its location in sort of high resolution or fine scale. And when I talk about fine scale, I normally talk with regards to like a location every second. And then also begin to know when they're doing that behavior to pick out diurnal patterns or similar. And then hopefully try and lead that into how we can use this information to better conserve the species. So I've been involved in designing, because our lab, to be fair, it utilizes a range of ways to collect this data. So not only do we work with commercial companies to use our technology alongside commercial collars, but we also build our own collars from the ground up. And it's not always collars. We've used backpacks, we've used different types of glues, or we just use simple tape for some bird species. But for this example, because I'll be working on prairie dogs and ferrets, is a collared example, and the projects or this project I'm associated with, these are the kind of design I've got in mind. So we've got a Atlas logger or a GPS logger, which isn't developed at Swansea. That's sort of a different company, and that would obtain us a location using some sort of triangulation with base stations or satellites. And then I'll go into this in more finer detail. We've got the daily diary. So this was developed at Swansea University. I'm under the impression that Swansea was one of the first animal labs to utilize accelerometry data on animals and also use magnetometers. Now everyone else is sort of caught up, but we're still, by being there first, we're still almost on the cutting edge of new analysis and also our new lab built software and new approaches of how we use this high-res data. So the daily diary collects acceleration in 3 axes. So that's the Z or the heave, that's the up and down motion. And you've got the X, which is usually the surge, which is moving back and forward. And then you've also got the Y, which we refer to as the sway, which is like side to side. So you're getting that motion in 3 axes. And then we also collect magnetometry in 3 axes. And we are able to collect some environmental data as well using sensors on the circuit board. So mainly we normally collect temperature and pressure. I think it's always nice to understand what the sensors are actually doing so that we know that there's not any sort of magic going on here. These electronic pieces of equipment are being almost stimulated by the animal. So an accelerometer is a tiny piece of equipment. It's literally, you couldn't even try and quantify its size, but you can just about see it to eye on the circuit board. And it involves a fixed plate, as you can see here, and a plate that's almost like a finger-like structure, which is situated in a spring. And then when the animal moves, it's able to stimulate that motion, and then currents run through those plates, and however the current changes is able to quantify that movement, or what we call it as acceleration. And that's acceleration G. That's quite key to understand, because sometimes people, we're not talking about true speed here. We're talking about G, like you feel in a car when you turn, or maybe in a plane. And but we are able to obtain that acceleration. And when we know the mass of the animal is constant, we can use some simple physics to actually derive the forces involved, or the forces that animal is pulling based on that data that we collect. And also that data can be analyzed in a range of ways, which I'll go into during this talk. And I'm not a magnetometer expert, but we effectively collect magnetometry in 3 axes, because if you imagine magnetic north in one direction, if you tilted upwards, the readings you would get would actually change. So we can use that magnetometer to derive orientation, but fundamentally, hence the picture, we use it as a compass. So we're going to be able to derive heading. We're going to be able to derive the direction that the animal is facing in relation to the globe. I hope I haven't lost you so far, because I understand I'm just throwing massive loads of information at you, and whatever sticks, stick. But it'd be really nice if this nugget of information sort of stuck with you throughout the talk. And it's the one acronym I'll probably end up using a fair amount, and it's called VEDBA, which stands for Vectoral Dynamic Body Acceleration. But effectively, to keep it in layman's terms, you take into consideration all the acceleration data in all 3 axes, and it gives you a metric. It gives you 1 unit considering all those 3 directions. And it's effectively activity. So it shows you the activity of the animal, because the device is on the animal, and the animal's movement stimulates that sensor, and therefore gives you the different measurements. And but it's worth noting that that metric can then be calibrated, or it gives you a good proxy for energy expenditure. And there's been a fair few amount of studies done on this, looking at different, is it being to calibrate the sort of that metric to associate it with energy expenditure for a range of species. I think we're led to believe it works better for like warm-blooded animals. And then VEDBA can also have a good relationship with speed. So I say a good relationship because there can be lots of variation involved. The relationship is strong, but like I say, the variation can be quite big depending on the animal's walking gait, depending on the superstrate, so what the animal's walking against, depending on the substrate, what the animal's walking on, and then it might even change with the weight of the individual, and obviously it's going to change across species. And I spoke about all these things because the thing I ended up specializing in during my PhD is a process called dead reckoning. It's a sort of nautical term where you can take the direction an animal or a ship, if we're talking nautical, is facing, and then we can work out its speed and therefore derive its distance, and step by step reconstruct where the path of an animal has been. And the reason I've explained that is because we can take our vedba as our proxy for speed to derive the distance, which comes from the accelerometry. We can take the mag, which is going to give us the heading, and we can then take a low resolution GPS path because we can able to undertake this analysis from one GPS point to another. So the reason for this is, although dead reckoning performs really well at high resolution across a large scale of data points, actually, because we're collecting this data at very high resolution, which I should have probably mentioned earlier, we're collecting it at 20 hertz. We're collecting 20 times per second for using these daily diaries, and that's minimum. That's what we normally use for terrestrial species. When we get up to more where we get to birds, where they're able to change the direction and the dynamics of their movement alter so much in splits of a second, you're looking at 40 hertz or even 120 hertz or even 200 hertz in some cases, because it really raises the quality of the data. So you're doing so many calculations step by step to give this high resolution path that these errors can catch up with it and the path can begin to drift. But then we're able to take our GPS information and fix any error that accumulates over time. So effectively, you take your low resolution GPS path, for example, once every 30 minutes or once every hour, and then you could dead reckon between the interpolated 2 points to give you a high resolution path. So you could have a 20 hertz location. I mean, for many species, you don't need to know the animal's location 20 times per second. But for example, here I've got a goat, which was dead reckoned for 24 hours, and that's 2 hertz. So it's giving you 2 locations per second. And it's really interesting what Brett was mentioning before, because these daily diaries need a fraction of the battery consumption, or their battery life is much larger compared to the size of the battery, because it has, unlike GPS, it doesn't need to communicate with satellites. It's all kept internally. So it's best to start to give you examples. So I mentioned penguins before. This is a penguin here, and the big benefit of dead reckoning is it also can perform when the GPS can't, because GPS locations, for example, commonly collected, it can't pass through salt water or thick tree canopy, and also GPS can be fairly inaccurate at times. So here we have a dead reckoned penguin, but the blue sections show where the GPS is taking over. So this is the bird at the surface of the water. And then as soon as it goes down to dive, this is utilized by the sort of straight line as the GPS is interpolated. The GPS drops out, giving you this straight line, but the dead reckoning takes over and gives and starts to give you a more detailed picture, a better idea of the actual sort of space the animal is using. And you wouldn't be able to obtain this data any other way apart from using dead reckoning, to my knowledge. And if we even look at these, this dive, for example, you've got this amazing 3D movement of where the animal goes down, it intercepts prey. So the tortuosity of the path starts to change and wiggle. And this is commonly accepted that a dive wiggle in depth signifies a prey pursuit. And it pursues the prey, obtains the prey at some point during that tortuous path, and then begins to sort of peruse for longer periods during the bottom phase, and then it ascends again. And it does this sort of circular motion as well, almost as it circles the shoal of fish. And this amount of detail would not be able to be collected if it wasn't for dead reckoning. We wouldn't be able to give that narrative without that high resolution data that we collect at Swansea. And it's worth noting in this case where the penguin's been studied, or this range of species, but penguins to be studied so detailed within our lab, this has been calibrated with feeding sensors and stomach pumps and all sorts. So this isn't just sheer interpretation. Also, during my PhD, I worked on ungulate species in the Alps. And this is to show that my GPS data was collects once every 4 hours. And look at the amount of detail you're missing in that path from that low resolution. I mean, one fix per 4 hours is quite low resolution stuff, but the dead reckoning is able to perform throughout that and give us a better idea and give us high resolution and give us good location estimates. And you might be like, okay, well that's great. It's fancy stuff to collect sheer quantities of data, but does it actually have a purpose? And I would strongly argue, yes, of course. Not only within that path do you have these zigzags showing the movement strategy of the ibex, showing that it's undertaking different paths to mitigate the effects of the slope, but also there could be movement barriers here that we aren't able to interpret with the GPS alone, because you have your ibex moving here, and then for some reason it might be some sort of cost benefit motivating the animal to move away, or it could be a movement barrier. It chooses to completely displace its location compared to the least costly or the least time costly path and move all the way around. And what's nicely working here as well, the dead reckoning is really trying to work its best accuracy wise, and it's moving around these snow areas, which ibex have shown to avoid when they can. Next thing as well from my PhD, it gives you, this is an example which shows the sort of detail of behavior that not only dead reckoning path gives, but the acceleration data can give. So this instance actually is tragically one of the mouflons I tagged was killed by a wolf, and this was confirmed by post-mortem. And you can actually look at the final sort of pathway or the final few hours of the locations of that animal. So this is the one GPS available in blue. It's not very clear, to be fair. Hopefully you can see my mouse cursor, and the yellow is the dead reckon path. And it really gives you so much more detail, but more importantly, you get that final pursuit. It only lasts about, I think it lasts about 3 minutes, but that pursuit is really interesting, the dynamic movement involved. So you get this tortuous path as the mouflon tries to move side to side swiping, all this dynamic movement to avoid being caught. And then you have the acceleration data, which gives you an idea of the impacts and the forces involved. So we have this level is interpretation, because obviously we don't necessarily have the focals to prove it, because this happened late at night or in the early hours. And we have this pursuit where we had this systematic constant steps, but it was at speeds, and the walking gait was really high forces that we'd never seen previously in the data. You have these changes in posture and emotion, and these high impacts, which could have indicated the kill of the initial feed. And then you had this drop off, or this really still data afterwards, which was more than likely going to be the dead animal. I've also more recently worked in a wild dog project, tagging African hunting wild dogs and displaying some of this data here. So you've got one fix every minute, just for the sake of displaying purposes, because it's quite hard to display 1 Hertz data. And actually the resolution of the sort of path or the quality of the image doesn't really give you much more in the way of detail. And this was 1 fix per minute from that dead reckon path of 72 days data. And the kind of take home from this figure is the density it gives. So this has a nice story of this wild dog dispersed from its pack, started to roam its area, looking for food and potentially other new pack members. And it began to disperse into a different country, which the current conservation group did not want. So it led that animal back into semi-captivity. And if you had the GPS alone, you wouldn't be able to get that sort of detail. And here again, you're able to almost have this heat map effect with density to give you an idea of, if in this semi-captivity or captive pen, where is the animal spending most of its time. This is the same path, but you're able to map variables. So there's that metric I spoke about, VEDBA. So if we talk about it in regards to energy expenditure, it's exactly what you expect. So the sort of spaces between the dots give you an idea of speed as well, because that's almost time. And you can see when she undertakes these large, these sort of long distances, the VEDBA goes up, as you'd expect, because she's executing, you know, movement, she's displacing herself, which would cost a lot of energy. And then when she's waiting in locations, she's using much less energy, so therefore the VEDBA value is a lot less. And that really ties in nicely with your mean temperature as well, because during the cool periods, she's moving, she's choosing to move during cooler periods. And when it's hot, so this is the temperature taken by the daily diary, it's the temperature the logger is experiencing. When the log is hot or when it's hot in the day, she's choosing to not move so much. And you can then take daily means. And although, you know, this is high resolution data, you can mean out and get a more accurate mean, even that just to kind of display the data. And you can take daily mean temperature and daily mean VEDBA, and you can see that during the hotter days, she's much less active on average. Let's just move back a second, because obviously this kind of ties in really nicely, because the whole scope of this project was working with Professor Rosie Woodruff, was to understand how climate change might be impacting these wild dogs, how might high temperatures impact their daily activity. And as you can see, for the detail hasn't been unpacked yet, it's yet to, you know, we're yet to look into it, but this simple relationship already shows that for whatever reason, during higher temperatures, they're avoiding being active. So they've got a choice, realistically. Do they end up forcing themselves to become more active in hotter temperatures, so exposing them the risk of overheating, or do they choose to be less active, keep this relationship going, and therefore become less active and therefore obtain less prey? Also, talking less about dead reckoning, you can use the accelerometer data to quantify behaviors. So you can actually visualize, we use some lab-built software called Daily Diary Market Trace, and you can visualize your data in large quantities at 1 million or 2 million data points at a time, and look at how the animal's sort of accelerometry changes over time. And these behaviors or these sort of changes can be systematic, they can be regular. So you're able to quantify them. So you can look at an animal for X amount of hours, quantify some behaviors, and then with a degree of accuracy or a degree of sort of quantifiable accuracy, you can then see how your animal's behaviors, you can mark them across the whole time scale. So you take a few hours of observations and able to understand how, the rest of your data set. So you can look at days or months worth of data. And you can have rules which are very simple, which are really difficult for reviewers or to argue against, where you've got active and inactive, to the more complex, resting, grazing, moving. You've got this head clash or this rutting behavior, to the more dynamic, which you saw before, or more specific. You can really look at a one-off behavior, for example, this mouflon being pursued and being attacked. And it's worth noting, a lot of work has been done on machine learning, being able to quantify behaviors, but we have an approach where machine learning is an extremely powerful tool, but our lab-built software almost has an open box approach where you can visually inspect your data. You can identify the trends yourself, so you can explain them, rather than having a closed off approach where you kind of just let the computer rule. And then you can tie the dead reckoning with the quantified behaviors like I've done here. You have, thankfully, I worked on ungulate species, which 80%, 85% of the time just undertake the same 3 behaviors, where you have resting, grazing, and walking, and you can map that out to see where they're undertaking this behavior. I don't have this analysis here, but in my PhD, in my chapter, I wrote up how to identify what these domestic goats were actually selecting to graze on, which was really important within the Alps, because there was potential competition with the wild species, but yet farming was an important practice for the Alps. So that needs to be sustained, but it's almost like this approach of how can we better manage the farming so that both can live alongside one another. So yeah, you've got this distinct pattern, or maybe it doesn't seem that so distinct with the messy data. It's kind of to show the sheer quality, the quantity of information you get back. But the idea that the goats moved out to these grazing patches, so they're clearly selecting different habitat types to graze upon, also while grazing as they go. And then when they get to these patches, the grazing intensifies, and then they come back to this pen which they were kept in over some nights, but not all. And yeah, you can then start to compare that to the ibex species. So to begin to sort of get an idea of the, if you like, the thermo niche, like what sort of is affecting what areas they're able to operate in, or in regards to temperature in this instance. So we have this nice, we can look at the diurnal pattern of hourly proportion time spent on grazing. So this is the average for my 6 ibex species and my 30 goat individuals that I looked at. And you can see how that alters between the two species. But I think this is the figure on the right is sort of the more interesting, where I compared the hourly proportion of time spent on looking at how that compares to temperature. And you have this ibex being very strict with the temperature. It's choosing to graze less during those hotter periods, whereas the goat was less fussy. So domestic goats are fairly robust, or it's accepted that they're pretty robust. Hence, they've been utilized across the continents as a farm animal. And yeah, as you can see, the amount of, there was no relationship there with temperature. So i.e. the goat could graze whenever it wants, despite the diurnal trend, which could be a potential threat to the ibex species, because you have a more robust animal able to graze on certain patches and similar. I was able to quantify more complex behaviors. So this is a head clash behavior. So this is when the two individuals bash heads with one another. And this shows sort of the breakdown, how I used a time element to break down each section of that behavior and then quantify it. And then I was able to map it to show density. And it worked out really nicely in the end, because 3 of my individuals I collected data within the rutting season. Not the largest sort of sample size, but for a proof of concept, it works really well, where you have this rutting throughout, which the literature shows the ibex does. It sort of competes throughout the year to build a hierarchy system, and then makes a final push during the rutting season. I don't know why that says hour, that's meant to say month, but you always discover these things when giving the talk. And then we have this, distance moved. And once again, this really nicely lined up with the data, because ibex have been shown to, throughout the year, they forage less and less coming up to the rut as they begin to conserve energy ready to perform within the rut. And during the rutting period, the distance moved drops right off, so that they can save all their energy to compete for females. And more interestingly is you can actually map that out. And then we found the area use didn't actually change despite the distance moved dropping right off, showing that foraging was barely undertaken during the rut, but they were still operating in the same areas. And relating this directly to conservation, I think this has the best sort of relatable finding. You could, because it's such an important phase for the animal shown here, they're pushing themselves right on the sort of energetic limits during this period by not foraging, by saving all their energy for competition. But the Alps is commonly used for tourists throughout the year, because they have a snow season. They have in the summer, it's really popular for paragliding, walks, and all sorts, and has a thriving tourism industry. So understanding them conflicts and clashes and managing those is extremely important. For example, you could say, you can use this information, this detailed information, to say, right, during these months, do not use these areas, or keep them closed off, or limit them to tourists to prevent any sort of human disturbance, because human disturbance during this period could actually affect the fitness of the species. And finally, just going into some of my Masters of Research work, where I worked on a giant tortoise species, which was interestingly being used as an ecological proxy. So it was this island off the coast of Mauritius, and this island used to have a giant mascarene or Mauritian giant tortoise on it, which sort of helped disperse the seeds, helped to manage some of the weeds, but it went extinct. And sort of boldly, the Mauritian Wildlife Foundation actually used a proxy species, the Aldabra tortoise, and put them on that island. But for whatever reason, they weren't dispersing across the whole island. So we took some loggers, we tagged 30 tortoise of the, I think there was about 300 at the time, and to build an idea of where they're going, what areas are they using, and why might they not be necessarily utilizing the full island, because they wanted the species to have the impact across the whole island rather than small sections that they were selecting. The findings from the actual thesis, it ended up being, you know, they're not necessarily at full capacity, so the competition wouldn't necessarily make them disperse. But more recent work I've done on this showed that looking at VEDBA as a cost of transport, we were able to look how the cost of transport was changed depending on the slope when they were moving, to build this idea of if that is the biggest factor impacting the distribution of the species, because what would you call it, vegetation or what they were eating wasn't limited, then how might, if we map that out, can we begin to predict where the animals are likely to disperse to, and while also showing areas that might be inaccessible because the species weren't ever found at that sort of slope or incline. And yeah, I actually have later data to look at, because this is a prediction using the data I collected, but colleagues has collected more, and it looks like they are using these dark purple areas, which are shown to be less costly, which would make a lot of sense because they're likely to use areas that they're going to spend less energy in. So overall net, they make a energetic profit. And the 3 take-home messages kind of showing you how, you know, talking about our lab built collars while also working with commercial companies, and analysis of this data can give you an idea of the what. So the behaviors, you can able to quantify a range of behaviors depending on the quality of data and other data you have available. The where, you can use dead reckoning paths to take your low resolution GPS data to map out those behaviors. And the when, you obviously have an internal clock clip there, so you can look at these different patterns of behaviors and activity budgets. And this information can really be utilized by conservationists so they know where their animal's going, what they're doing in much, much finer detail, unlike some of the sort of low resolution stuff available at current. And yeah, special thanks to a range of people, including people at Swansea University, but also the ZSL, NERC, which was associated with the Wild Dog Project I worked with, and then during my PhD, Grenoble University, and then during my master's, Durrell and the Mauritian Wildlife Foundation. And yeah, any questions?
Thank you, James. I had a quick question about, do you want to talk briefly about the black-footed ferret and prairie dog work you guys are doing with EULA? Yeah, of course. I could have probably afforded to put a slide in there about potential plans, but the idea is we haven't collected any data yet. We're just working on designing the collars, and the reason I really drummed home the message about dead reckoning is we hope to do something that's not been done in this detail before, where we're hoping to obtain locations of the animals when they're underground, because obviously you're not able to obtain any locations when an animal is underground because you're not able to communicate with anything. Radio signals don't tend to like to pass through ground. So we're looking to map out the burrowing system of the black-footed ferrets or the prairie dogs, and yeah, go from there. And I don't think it's ever been done before to that level of detail. So and then from there we hope to show how the animal's behavior might be changing, how, well, I mean, as you've seen, the possibilities are endless. We hope to kind of collect the data and maybe let that ask the questions rather than the other way around. Excellent. And what exactly are you hoping to find from this? So like I've said before, so
the burrowing systems have never been mapped out before, and it's is where black-footed ferrets spend majority of their time. They do a massive re-release project, and you really want to kind of look at what behaviors are they undertaking. I think something that's kind of overlooked during conservation is the fine detail of the energetic budget. And I really want to see how much energy the animal is spending in regards to VEDBA, and then how much are they actually obtaining by quantifying hunts and feeds, because building that picture could be really important to understanding what might be going wrong with regards to the animal's fitness, et cetera, et cetera. And yeah, build up that fine detail. Yeah, so I think it's a bit overlooked. You're always looking at like, oh, we need to protect this habitat because of habitat destruction, but realistically, yeah, this whole sort of getting down to the nitty-gritty numbers of energetics can be overlooked.
Okay, we have time for one quick question. Brett, you want to go ahead? Oh, you're on mute.
Yes. Great talk, James. Thanks. Just as quick and probably easy question. So for linking the DBA to the known behaviors, did you have to do a lot of behavioral observation work, or did you just sort of assume behaviors based on what you know about the DBA profiles?
Yeah, cheers, Brett. Yeah, fair question. So for the examples I used today, I had observations. The only sort of leap I took is the ibex. I had limited observations with, so to test my rule, I used a surrogate species in the goats, which was quite a unique approach. And it is becoming more accepted within the biologging community to be able to just interpret the different readings and different signatures you are getting, but realistically observations are the way forward, and it's a sure way of ground truthing your quantification of rules. So it's always best, the more observations you have, the best it is. That is the sure way of doing it, and that's what I was able to do with grazing and walking and resting with my domestic species, because they're very accessible.
Great, thank you.
And there's one question in the chat for you, James. You can chat back to Chris. And if there are more questions, feel free to put them in the chat or ask James directly through the chat. We may have time to come back to them, but I wanted to give Peter a chance to also give a quick summary report from the grasslands monitoring meeting. And if we have time to discuss next steps.
Actually, I think that thanks, Lacey, and thanks to the speakers. Those were both fantastic talks. Actually, it might be better to just spend the time on those questions and then schedule the grasslands thing for the next meeting. We're still working on the report. I mean, I can't just spend 5 minutes here to update everyone on that. This is a meeting that happened on the 11th January. It was a one-day meeting to network between projects that work on grassland science and look for joint questions and think about how we could create more synergy and perhaps have a broader grasslands ecology conservation program. And the workshop was really cool. Actually, we had 20 speed talks, just showing the breadth and the depth of science that's happening on grassland species and grasslands issues that ranged really from genomic and genetics research on specific animal populations to remote sensing and landscape analysis and sustainability kinds of questions. And I think I'll leave it at this right now because we're still working on finalizing that report. And I think since we have so little time left, it might actually be much better spent bringing up what other items need to be discussed, but also maybe having more time with James and Brett.
Okay, that sounds great. Yeah, I think the only thing I'll point out, which maybe we'll resurrect when we have the grasslands follow-up, but there is a link in the agenda to a request for project ideas or on kind of a survey of ongoing grassland projects that involve animal tracking to either study connectivity, community dynamics, environmental change in some way. You know, I know we're a relatively small group and we may have we may know, I feel like we have a decent idea of what's out there, but I also feel like every time I speak to somebody, there's some great idea brewing that might be useful to at least raise awareness of, and talk about how it might plug into this broader grasslands monitoring effort. So if you would like to make sure that that project is included in the conversation, if you'd like to play a bigger role in thinking about how animal tracking might contribute to a grasslands conservation initiative or grasslands monitoring initiative, fill it out. It's really quick, and I'm sure you'll see it again if you're not quite ready to do that at this point. So
Just to,
yeah, go ahead.
Lacey,
Peter.
just to add to this, I mean, so there's really two pieces here. There's that grasslands meeting on the first day that was really about what are the big questions and well, what's the niche that we potentially have. And then after that, we had a longer conversation in the following days about indicators and what actually would look up, what are the kinds of things we would measure if there were a bigger project. And I feel like this movement thing really fits. It fits in both, but it would be nice to have an idea of what's going on and what people are planning. Movement didn't feature very strongly in that first meeting because it was very, very broad. So
I'm going to put a link in the chat to the survey. Okay, great. So with our last 10 minutes, we can pick back up with questions for James or Brett at this point.
I don't have a question, but it's just interesting how we're talking about grasslands, and this is a system where we're already tracking some key species. They're kind of there together, but they're not really overlapping. And I think, you know, the opportunity to actually simultaneously track different species, it's going to be a big challenge, I think. But we do have a place where we're already doing a lot of work, and they're almost adjacent. We've got swift fox on the Indian reservation. We've got bison on the AP lands, and meadowlark, curlew have been tracked. You know, they're all kind of there. Potentially there are going to be more with prairie dogs, black-footed ferrets, others. You know, it's but you know, how close and are they going to be? And they're not really overlapping. You know, maybe pronghorn at some point, but again, I don't know. It's kind of like just missing maybe this opportunity.
Yeah, that's part of why I wanted to have this meeting. I think is I feel like we're on the verge of being able to do something like this, but maybe we haven't all come together purposefully with that intention or identified these opportunities that are almost there to make that happen. So just trying to get our collective mind thinking in that direction. But go ahead, Peter.
yeah, I mean, I think there is thinking happening. I mean, I don't know whether Amy Johnson is here, but there's also, I think, a plan of tracking multiple species with this tracking network in Virginia landscapes or grasslands. I think that's more focused on birds, but those things could be expanded. I do have a question, which is really just Chris's question for James. I'd love to hear more about the kinds of algorithms or tools, James, you're using for the dead reckoning, whether you have any recommendations. I think many of us actually have very high detailed accelerometer data, and I think there's a big interest in, I don't know whether it's a big interest, but I know some people are interested, and it would be nice to hear a bit more about what tools there are and what tools you've used.
Yeah, so I put a link in the chat, and that's recently a colleague I worked with them too, and they've actually implemented into R, so you're able to run an R script, or it's not been turned into an actual package yet. You have to get his script and make a function from it, but it does rely on magnetometer data is what it requires, which is the unique thing about it, to be able to get that heading. The only other alternative was if you only had accelerometry data, is maybe you could interpolate across GPS, sync up the two, and then be able to get an idea of change of speed or change of activity along that interpolated line. But yeah, the magnetometer is quite key in getting that heading. But yeah, so sorry, there's a link in the chat, and that's the most up to date, and it gives all the calculations, and also I think it does supply the script somewhere. And if not, I can forward you to Dr. Richard Gunner, and he can give you the script himself, I'm sure.
Great.
Anybody else?
I can just say that, at least in Montana, to what Mel said, we've been talking now for a few years with Jared and Lacey about like a movement of life field laboratory where we can tag a bunch of species at the same place at the same time. And of course, it's a huge challenge. We tried to like play around with like a LoRa network, didn't really work. We're back to more of satellite type of tags right now, but I do think that there is potential now, working with James, using this lower resolution GPS tags together with the dead reckoning, which can really open up a whole new world for us in terms of looking at species interactions, especially us thinking more about the ungulates in that region. But to something that Bill also pointed out in terms of what resources are they using and when. So for example, with Claire Bresnan, who is a PhD student working on bison movement, she's on the call today. So she's combining a bunch of methods to try to understand how bison are impacting vegetation and other ungulates. So incorporating also camera traps, collecting vegetation samples, and also using high resolution drone imagery. So it is a ton of work, but I think it can only be done when you combine all of these methods together to get to these questions. But I'm not exactly sure how we would scale it up to much bigger regions, but something to explore in the future.
I like that. I mean, I think what I guess, you know, my question would be, I think it would be great to do multiple species, but what's the question, right? Is the question whether interaction complexity is different, you know, between more naturally managed grasslands and heavily grazed grasslands? I think it would be really worthwhile thinking that through, and then also thinking about which species would be the pieces to the parts where it would be really interesting to do that. Yeah, I don't know. I think the challenge is a little bit like, okay, what is a sweet spot for a model that we could kind of try to use to address some interesting questions and also really broaden ecological theory at the same time. I think that would be really cool.
Well, this has been a really enlightening meeting for me, and I really appreciate James and Brett's time and effort to share your work with us. I think they've both been have a lot of relevant themes to pushing forward ideas that we want to be working more towards. And yeah, so I'm sure there will be follow-ups with both of you for just more questions, more potential collaborative opportunities. And as Peter said, we'll carry on the grasslands conversation within Smithsonian in the next few weeks and months. And thank you guys for joining, sharing your time.
Thanks,
Oh.
Lacey.
Yeah.
We're
Thank you for
We're going to be
organizing
thinking
it.
about you, so
Yeah,
Thank
keep us
you.
updated.
I will, yeah. See
and
you in
thanks for
August.
organizing another really excellent meeting. So that's really cool.
Yeah,
You guys
sweet
make
you,
the last
everyone.
bit of your time.
Thank you.
Those are great talks.
Right.
Good to see everybody. Thank
All
you.
right.
Bye.
Thanks, everyone. Bye.
Thank
Bye.
you all.
Bye.